shape either smooth or wrinkled, seed color yellow or green, flower color purple or white. There wasn't this confusing spectrum like, say, human height.
Right.
This meant he could cleanly classify the hundreds, thousands of plants he analyzed.
And it wasn't just how they looked, their life cycle helped too.
Yes, definitely. They have a short generation time.
Yeah.
That allowed him to track multiple generations relatively quickly.
And he needed that F2 generation, right,
Yeah.
to see things reappear.
He absolutely did. To see those recessive traits pop back up,
Hmm.
and maybe most critically, he had complete control over their reproduction.
How so?
Well, he could easily let the flowers self-pollinate.
Mm.
Or he could step in and do very careful, controlled cross-pollination by hand, transferring pollen with a brush. This level of control, plus his dedication to using really large sample sizes, that's what gave his results the statistical firepower, the reliability, to establish what we now call universal laws.
It's just amazing those statistical laws still hold up. So, okay, to move forward, we need to get the language right. The lexicon that came out of his work, even though the word gene itself came much later.
Right. Let's lay out the basics. A gene, as we define it today, is a heritable sequence of DNA that codes for a specific product, usually a protein, sometimes an RNA molecule.
And its address on the chromosome.
That specific physical location is called the locus.
Okay. And the different versions of that gene at that locus are alleles.
Right. Alleles are just alternative versions of the same gene. And we use a standard notation to keep track. A dominant allele gets a capital letter, like A, and a recessive allele gets the lowercase a.
And we get one from each parent.
We do. So when we look at an individual's genotype for that gene, we look at the pair of alleles they inherited.
So if you get two dominant ones, that's homozygous dominant, AA.
Correct. And two recessive alleles makes you homozygous recessive, AA.
And the one that reveals the most about how these interact is the heterozygous state.
One of each, exactly. And this is where we make that crucial distinction between the genotype, that specific combination of alleles, the letters like A, and the phenotype.
The observable outcome.
The observable trait, yeah, like a purple flower, or in humans, maybe brown eyes. These terms, they're the essential building blocks for really understanding Mendel's three core laws.
Okay, let's tackle law number one, the principle of segregation. What's actually happening here?
So the rule basically says that any given trait, the two alleles an individual has, they separate from each other when that individual makes their gametes.
Their sperm or egg cells.
Right. It's a fundamental event during meiosis, the cell division that produces gametes.
And the result is that each sperm or egg cell carries only one allele for each trait.
Which is the core reason sexual reproduction works the way it does. The significance is huge. It explains why offspring get just one copy of each gene from mom and one from dad. It guarantees genetic diversity. And crucially, it also solved that mystery that puzzled earlier naturalists. Why could a trait, especially a recessive one, vanish completely in one generation?
The F1 generation, the first cross?
Yes, the F1 hybrids.
Oh.
And then reappear totally unchanged in the next generation, the F2s.
The recessive allele wasn't gone, it was just hiding.
It wasn't destroyed or blended, exactly. It just segregated away from the dominant allele during gamete formation and was masked, its effect hidden in the heterozygous F1s.
Okay, that makes sense for one trait, but law 2, the law of independent assortment, ups the complexity. It tracks two or more traits at the same time.
Yeah, this law states that the alleles for different traits are inherited independently of one another. So the inheritance of, say, seed color doesn't influence the inheritance of seed shape.
Meaning they aren't physically linked when the gametes are forming?
Generally, yes. The reason is usually physical. In the cell, the genes for different traits are often located on different separate chromosomes.
Ah, okay.
And during meiosis, these different chromosome pairs line up randomly before they separate. Mendel actually confirmed this using his dihybrid cross experiments.
Right, that famous cross where he took plants that were heterozygous for two traits, let's say yellow-green color, YY, and round wrinkled shape.
Exactly. He crossed YR with YR. And he didn't just get the combinations he started with, yellow round and green wrinkled. He also got new combinations, yellow wrinkled and green round.
And in a specific ratio.
Yes, that classic 9.3.3.1 phenotypic ratio. 9 showing both dominant traits, 3 showing the first dominant and second recessive, 3 showing the first recessive and second dominant, and 1 showing both recessive traits. That ratio is the mathematical signature proving the two genes assorted independently.
And this is a really important scientific qualification, right? Something Mendel couldn't have known back then because he didn't know about chromosomes.
That's the critical piece of nuance, yes. The law of independent assortment only holds true if the genes are on different chromosomes. Or if they happen to be on the same chromosome, but they're located very, very far apart from each other.
Why does distance matter?
Because if they're physically close together on the same chromosome, they tend to be inherited together as a block. They're physically linked.
So they don't assort independently.
Right, they travel together, violating that 9.3.3.1 prediction, we call that phenomenon, linkage. And it becomes super important when we talk about gene mapping later on.
Got it. Okay, and then law 3, the law of dominance. This one seems maybe the most straightforward, the one we see most easily.
It's basically the principle of masking.
Yeah.
In a heterozygous individual, that AA individual,
Mm-hmm.
the dominant allele, the A, is the one whose trait gets expressed in the phenotype. It completely hides the presence, the effect of the recessive A allele.
And that explains why in Mendel's first crosses, that F1 generation only showed the dominant trait.
Exactly. All the F1s were heterozygous, and the dominant allele masked the recessive one.
The real genius here, though, wasn't just seeing these patterns, it was predicting them. His work showed, fundamentally, that genetics is a game of probability.
Absolutely. And we use basic rules of probability, rules from mathematics, to predict these outcomes. If you know the genotypes of the parents, you can actually calculate the expected ratios of genotypes and phenotypes in the offspring.
So what are the tools in this probability toolkit? First up is the multiplication rule.
Okay, you use the multiplication rule when you want to find the probability of two or more independent events happening together. Think and D events.
Both this and that happening.
Yes. So the probability of event A happening and event B happening is found by multiplying their individual probabilities. PA and BPA times PB.
Can you give a quick genetic example?
Sure. Let's say you want to know the probability of a child inheriting a specific recessive allele from parent 1. If the parent is heterozygous, that probability is 12. And what's the probability of inheriting that same recessive allele from parent 2, assuming they're also heterozygous?
Right.
Also 12. So the probability of inheriting both being homozygous recessive A is 12 times 12, which equals 14.
Makes sense. And the other main tool is the addition rule. This one is for OR events.
Right. The addition rule comes into play when there are multiple mutually exclusive ways to get a single outcome. If you want the probability of outcome A or outcome B happening, you add their individual probabilities. PA or B equal plus PB.
Mutually exclusive meaning if one happens, the other can't.
Exactly. For example, think about the phenotype of that heterozygote, AA.
Yeah.
What's the probability of getting the dominant phenotype? Well, you could have the genotype AA or you could have the genotype AA.
Right.
Those are two different genotypes, mutually exclusive, but they both result in the same dominant phenotype. So if you calculate the probability of getting AA, which is 14 from our previous example, and the probability of getting AA, which is 12, you add them together. 14 plus 12 equals 34. So 34 of the offspring will show the dominant phenotype.
And these two simple math rules, they're still the engine behind predictive medical genetics today.
Absolutely fundamental. Punnett squares are basically just visual representations of these probability rules.
Okay, so Mendel gave us this statistical blueprint, the basic grammar, but as we start looking at the real world, at actual biology, we quickly see the genome is less like a simple light switch and more like a really complex interconnected machine.
That's a great way to put it.
So we have to move beyond simple Mendelian patterns now and explore these more complex inheritance patterns.
Definitely. While Mendel's laws are foundational, they actually describe a minority of inheritance patterns when you look across all traits. Most genes show some kind of deviation from simple dominance.
Okay, let's start by really pinning down the difference between incomplete dominance and codominance. They sound similar.
They do, but there's a key difference. In incomplete dominance, the heterozygote, the AA individual, shows a phenotype that's intermediate. It's like a blend, visually speaking.
The classic example.
Snapdragon flowers. You cross a true breeding red flower, let's say RR, with a true breeding white flower, WW. The heterozygous offspring, RW, aren't red or white, they're pink.
A blend.
A blend at the phenotypic level. But importantly, the alleles themselves, R and W, are still discrete units. If you cross two pink snapdragons, you'll get red, pink, and white offspring back following Mendelian segregation rules. The alleles didn't actually blend.
Okay, so the units stay separate, but the expression results in an intermediate look. How does that differ from codominance?
In codominance, both alleles are fully and simultaneously expressed in the phenotype. Neither one masks the other, and you don't get an intermediate blend. Both traits show up.
And the go-to example here is the human ABO blood group system.
It's the perfect illustration. You have alleles for the A antigen, IA, and the B antigen, IB. These two alleles are co-dominant with each other, meaning if you inherit both, one IA from one parent and one IB from the other, your blood type isn't some intermediate type. Your red blood cells actually express both the A antigen and the B antigen on their surface. You have type AB blood. Both genetic instructions are fully executed and visible.
And the ABO system also introduces another concept, right? Multiple alleles.
Yes, exactly. While any one person can only carry two alleles for the ABO gene, one on each homologous chromosome, within the human population, there are actually three common alleles circulating: IA, IB, and the recessive allele I, which codes for no antigen.
So more than just two options exist in the gene pool.
Right, which allows for more genotype combinations and the different blood types we see, A, B, and O, which is genotype 2.
Okay, now we're stepping into even more complex territory. Interactions between different genes and interactions between genes and the environment. Let's start with pleiotropy.
Pleiotropy is fascinating. It kind of flips the usual one gene, one trait idea on its head. Here, one single gene ends up influencing multiple, often seemingly unrelated phenotypic traits.
How does that happen?
Often, the protein product of that single gene plays a really fundamental role in the body. Maybe it's used in different tissues or biochemical pathways. So if that one gene is defective, the problems can cascade across several different organ systems.
And the classic, though tragic, example is sickle cell anemia.
It is a single point mutation, just one DNA-based change in the HBD gene, which codes for part of hemoglobin. That's the root cause.
But the effects are widespread.
Incredibly so. That one molecular change leads to sickle-shaped red blood cells, which causes pain crises, chronic anemia, increased risk of stroke, damage to organs like the spleen and kidneys, but also, interestingly, provides some resistance to malaria in heterozygotes. One tiny gene change, massive diverse phenotypic consequences.
Wow. Okay, next up, epistasis. This sounds like genes bossing each other around.
Huh. Not a bad way to think about it.
Yeah.
Epistasis describes a situation where one gene essentially masks or modifies the expression of a different gene. It's about genetic hierarchy.
So it's not just additive effects, it's one gene acting like a switch for another.
Exactly. It's a non-additive interaction. One gene, called the epistatic gene, controls whether the second gene, the hypostatic gene, gets to show its effect. They often act sequentially in a pathway.
Let's use that famous Labrador Retriever coat color example to make this really clear.
Perfect example. In labs, there's a gene, the B gene, that controls the type of pigment produced. Black pigment B is dominant to brown pigment B.
Okay.
But then there's a second gene, the E gene, which controls whether that pigment actually gets deposited in the fur. This E gene is epistatic to the B gene.
It's the switch.
It's the switch. If a dog inherits two recessive alleles for this deposition gene, so it has the genotype E, it doesn't matter what its B gene alleles are. It could be BB, genetically black, or BB, genetically brown.
If the pigment can't be deposited.
The dog ends up yellow. The E genotype prevents pigment deposition, masking the effect of the B gene entirely. That's epistasis. The E gene's expression dictates whether the B gene's expression is even visible.
That really clarifies it. It shows genes don't work in isolation. They're part of these complex networks.
Absolutely.
Which leads us nicely to the idea of many genes working together on one thing, polygenic inheritance.
Right, this is kind of the opposite of pleiotropy. With polygenic inheritance, many different genes, sometimes dozens or even hundreds, contribute cumulatively, often in small, roughly equal ways, to a single phenotype.
And the traits involved are usually things like height, skin color, maybe intelligence.
Those are the classic examples, yes. Traits that don't fall into Mendel's neat, discrete categories like purple or white.
Instead, they show continuous variation.
Exactly. When you have lots of genes each adding a little bit to the outcome, the distribution of that trait in a population doesn't look like simple ratios. It naturally forms a smooth spectrum, often resembling a bell curve.
This is the realm of quantitative genetics.
It is, analyzing traits that show continuous variation.
And this is where we run into that concept that's often tricky and sometimes misused. Heritability, usually written as H squared.
Ah yes, heritability. It's frequently misunderstood. People often think it means, say, if height is 80% heritable, that 80% of your specific height comes from your genes and 20% from environment.
But that's not quite right.
That's incorrect, yeah. Heritability is a population statistic. It's defined as the proportion of the variation we see in a trait across a population that can be attributed to genetic differences among individuals in that population.
So it's about the differences between people, not the proportion within one person.
Precisely. If height has a heritability of 0.8 or 80%, it means that 80% of the height differences observed between people in a particular population living in a particular range of environments are due to the genetic differences between them.
It doesn't mean environment is unimportant.
Not at all. You could take a population with high heritability for height, put them all on a severely restricted diet, and everyone's height would decrease. The environment profoundly impacts the actual height achieved. Heritability just tells us how much genetics contributes to the variation around the average, within the range of environments studied. Environment always adds plasticity, setting the potential range for the phenotype.
Which brings us directly to that environmental influence. Genes don't operate in a bubble.
Never. Gene expression is incredibly dynamic. It's constantly being tweaked, modulated by external conditions. The sources bring up a really striking visual example. The Siamese cat.
Ah, with the darker fur on the ears, paws, tail, the cooler parts of the body.
Exactly. That pattern is a direct result of environmental temperature influencing gene expression.
How does it work?
There's an enzyme involved in producing the dark melanin pigment. The specific allele this cat breed has produces a temperature-sensitive version of that enzyme. It only folds correctly and functions properly at slightly cooler temperatures.
So in the warmer core body,
The enzyme is largely inactive, resulting in light-colored fur. But on the cooler extremities, the ears, face, paws, tail, the enzyme works, producing the dark pigment. The environment is literally switching gene product activity on or off in different parts of the body.
okay, so we have Mendel's basic rules. We have these complexities like incomplete dominance, codominance, epistasis, polygenic traits, and the environment weaving through it all. But how did we make the huge leap from Mendel's abstract factors, these statistical units, to knowing they were actual physical things on chromosomes?
That connection, that was a major breakthrough in the early 20th century. Mendel's work was actually lost or ignored for decades, then rediscovered around 1900. But it took the work of people like Thomas Hunt Morgan, primarily working with fruit flies, Drosophila melanogaster, to provide the concrete physical evidence.
Fruit flies. Why them?
Well, like peas, they were great model organisms. Short generation time, lots of offspring, easily observable traits, and crucially, they only have 4 pairs of chromosomes, making things a bit simpler to track.
And what did Morgan show?
Through meticulous breeding experiments, tracking traits like eye color, which turned out to be linked to the sex chromosomes, Morgan and his team definitively proved that Mendel's discrete factors were indeed genes located at specific positions on chromosomes.
This became the chromosomal theory of inheritance.
Yes. It grounded Mendel's abstract laws in physical reality. And if you look closely at the mechanics of cell division, specifically meiosis, the process that creates sperm and egg cells,
It matches Mendel's laws perfectly.
it provides the physical explanation. The separation of homologous chromosome pairs during meiosis. That's the physical basis for Mendel's law of segregation. And the random way these different pairs line up before separating explains the law of independent assortment for genes on different chromosomes. The cellular machinery validates the statistical patterns.
Let's zoom in even further now to the molecular level. Those alleles we write as A and A. What actually makes one dominant and the other recessive when we look at the DNA and proteins?
Okay, so alleles, remember, are just slightly different versions of the same gene, meaning their DNA nucleotide sequence is slightly different. Typically, what we call a dominant allele codes for a functional protein. Maybe it's an enzyme that works correctly, or a structural protein that does its job.
And in the heterozygote, having just one copy of that functional allele is often enough.
Usually, yes. Having one working copy of the gene often produces enough functional protein to result in the dominant phenotype. We call this haplosufficiency sometimes.
So what about the recessive allele then?
The recessive allele usually arises from a mutation that causes a loss of function. The mutation might result in a protein that doesn't fold correctly, an enzyme that doesn't catalyze its reaction, or sometimes no protein being made at all.
And because the dominant allele is providing enough function?
The defect caused by the recessive allele is hidden or masked. This really reinforces that central pathway of biology. The genotype, the DNA sequence, is transcribed into RNA, which is translated into a protein. The protein's activity, or lack thereof, leads to the observable phenotype.
Earlier, you mentioned linkage as the key exception to independent assortment genes close together on the same chromosome tend to stick together. How did scientists figure out how to use this to actually map gene locations?
This involves the fascinating process of recombination. So yes, genes located physically close together on the same chromosome are called linked genes, and they tend to be inherited as a single unit.
Not always.
Not always. During prophase meiosis, something remarkable happens. The homologous chromosomes pair up, and they can physically swap segments. This process is called crossing over.
Like shuffling parts of the chromosome pair.
Exactly. And when crossing over happens between two linked genes, it creates new combinations of alleles on a single chromosome combinations that weren't present in the parent. We call these new combinations recombinants.
So crossing over can break up linkage groups. And does how often it breaks them up tell us something?
Precisely. That was a genius insight. The frequency of recombination between two linked genes is directly proportional to the physical distance separating them on the chromosome. If two genes are very close together, the chance of a random crossover event happening right between them is low. So they'll mostly be inherited together, and the recombination frequency will be low.
And if they're far apart?
If they're far apart on the same chromosome, there's much more physical space between them, making it much more likely that a crossover event will occur somewhere in that region, separating them. So the recombination frequency will be higher.
So by measuring how often linked genes get separated in the offspring?
You can deduce their relative distance apart. Alfred Sturtevant, one of Morgan's students, realized this and defined a map unit, or centimorgan. A 1% recombination frequency equals 1 map unit of distance between genes.
Wow.
This principle formed the basis of creating the first linkage maps, showing the linear order of genes on chromosomes. It was foundational for all subsequent genomic mapping, even today.
But we know now the genome isn't just the static string of genes. The regulation, how genes are turned on and off, is incredibly complex. It goes way beyond that simple central pathway picture.
Oh, massively so. We absolutely have to consider the regulatory landscape. Traits don't just emerge from single genes acting alone. They arise from intricate gene networks.
Networks involving what kind of players?
Layers upon layers of control. You have transcription factors, proteins that bind to specific DNA sequences to activate or repress gene transcription. You have enhancer sequences, DNA regions often far away from the gene that boost its activity, and silencers that dampen it. And then there's the whole structure of the DNA itself, the chromatin states.
How tightly packed the DNA is.
Exactly. Whether the DNA is tightly wound around histone proteins, making it inaccessible, or loosely packed and open for transcription, that's a major level of regulation.
And it's not just proteins regulating things, right? There are RNAs involved, too.
Yes, the discovery of non-coding RNAs has added another huge layer of complexity. Things like microRNAs, miRNAs, and long non-coding RNAs, LNC-RNAs. They don't code for proteins themselves.
But they control the proteins that are made.
They often do, yeah. For instance, miRNAs can bind to messenger RNAs, mRNAs, and either trigger their degradation or block them from being translated into protein. They act like fine-tuning knobs, adjusting the output of many different genes.
And this brings us to something that really challenges the idea of the genome as just a fixed sequence passed down. Epigenetic inheritance.
Right. Epigenetics literally means above or on top of genetics. It refers to modifications to the DNA or its associated proteins that change gene activity without altering the underlying DNA sequence itself.
What kind of modifications are we talking about?
The most studied are DNA methylation, adding a small chemical tag, a methyl group, directly onto the DNA, which often acts to silence the gene nearby, and histone modifications, adding various chemical tags to the histone proteins that DNA wraps around, which can either loosen the chromatin, promoting gene expression, or tighten it, repressing expression.
So it's like putting chemical sticky notes or bookmarks on the genome, telling the cell which parts to read and which to ignore?
That's an excellent analogy. They change the interpretation of the genetic blueprint without rewriting the blueprint itself.
And the really mind-bending part, these marks can sometimes be inherited.
That's the crucial finding, yes. The sources confirm that in certain cases, these epigenetic marks, patterns of methylation or histone modification, can survive the process of gamete formation and be transmitted to the next generation.
Meaning the parents' experiences could potentially influence the gene expression of their offspring?
Potentially, yes. It suggests a mechanism for inheriting information about, say, environmental exposures or parental metabolic state, completely independent of changes to the ATCG sequence. It's a whole other layer of inheritance beyond Mendel.
Okay, let's shift gears now and look at the direct, sometimes life or death applications of these principles. Clinical genetics. Understanding those basic Mendelian ratios is still absolutely vital today, right?
Absolutely critical. Mendelian laws are the anchor for so much of modern medical genetics. They allow doctors and genetic counselors to calculate risks, predict carrier status for recessive diseases, and advise families.
So let's quickly recap the main patterns we see in human genetic disorders, starting with autosomal dominant AD ones.
Right. Autosomal means the gene is on one of the non-sex chromosomes. Dominant means you only need to inherit one copy of the faulty allele to have the disorder.
So it often appears in every generation.
Typically, yes, assuming people reproduce, and it affects males and females roughly equally. Classic examples include Huntington's disease, caused by those CAG repeats in the HTT gene, leading to neurodegeneration,
Others.
Marfan syndrome, a connective tissue disorder due to FBN1 gene defects, and one of the most common genetic diseases, autosomal dominant polycystic kidney disease, ADPKD, caused by mutations in PKD1 or PKD2.
Then we have autosomal recessive AR disorders. Here you need two bad copies.
Correct. You need to inherit the faulty allele from both parents to have the condition. Parents are usually unaffected carriers, heterozygotes. This is where carrier screening becomes really important for family planning.
And major examples here.
Cystic fibrosis is a big one, caused by mutations in the CFTR gene affecting a chloride channel, leading to thick mucus. Sickle cell anemia, which we discussed earlier, HBB gene, phenylketonuria, PKU, and inability to metabolize phenylalanine, PAH gene, and various forms of albinism affecting pigment production like TYR or OCA2 genes.
And finally, the X-linked recessive XLR pattern linked to the X chromosome.
Right. The gene is on the X chromosome. Since males only have one X, they're XY. If they inherit a faulty recessive allele on that X, they will express the disorder.
Females usually have a backup copy.
Exactly. They have a second X chromosome, which usually carries a functional copy of the gene, so they are typically unaffected carriers, though they can pass the allele on. This leads to the pattern where XLR disorders are much more common in males.
Famous examples.
Hemophilia A, factor VIII deficiency, causing bleeding problems. Duchenne muscular dystrophy, DMD gene defect, causing severe muscle wasting. And the common forms of red-green color blindness, opsin gene mutations.
The sources also dig into some specific mechanisms that add even more complexity within these Mendelian patterns.
Yeah, for example, achondroplasia, the most common form of dwarfism. It's autosomal dominant. But interestingly, it's often caused by a specific gain-of-function mutation in the FGFR3 gene.
Gain of function. So the mutation makes the protein too active.
Exactly. Unlike many recessive mutations that cause a loss of function, this specific FGFR3 mutation makes the protein constantly active, which inappropriately signals to stop bone growth. So even one copy of this hyperactive version is enough to cause the condition.
And things like Fragile X syndrome.
That's another fascinating one. It's technically X-linked dominant, though expression can vary, caused by an expansion of CGG nucleotide repeats in the FMR1 gene on the X chromosome. What's remarkable is that this repeat region is unstable. It can actually grow larger when passed down through generations, particularly through female carriers. This dynamic mutation leads to increasing severity in later generations. It breaks simple Mendelian expectations.
So these principles of inheritance, they don't just explain disease, they explain how organisms are built in the first place, the architecture of life. Let's touch on those developmental master genes, Hox and Pax genes. This is Evo-Nevo territory.
Evolutionary developmental biology, yeah. These genes are like the master architects or conductors of development. Hox genes are famous for setting up the basic body plan along the head-to-tail axis. They determine segment identity, essentially telling a block of cells you're going to become part of the thorax or you're part of the abdomen.
And they have that amazing collinear expression.
They do. The order of the Hox genes lined up along the chromosome remarkably mirrors the order in which they are turned on along the developing embryo's body axis. Gene 1 affects the head, Gene 2 the next segment, and so on. It's incredibly elegant.
And mutations in Hox genes can have really dramatic effects.
Oh, yeah. They can cause homeotic transformations, where one body part develops looking like another. The classic example is in fruit flies, where a mutation might cause legs to grow where the antenna should be, or an extra set of wings to develop. It shows how changing these master regulators can lead to large-scale structural changes.
And Pax genes, what's their role?
Pax genes are another family of crucial developmental regulators, particularly important for the formation of specific organs and tissues. The most famous is probably PAX6,
The eye gene.
often called the master control gene for eye development. It's required for eye formation across an incredible range of animals, from flies to mice to humans. Mutations in human PAX6 cause conditions like aniridia, where the iris of the eye is missing or incomplete.
And the big takeaway from Hawks and Packs.
It's the deep evolutionary conservation. The fact that fundamentally the same sets of master control genes are used to build vastly different animals highlights our shared ancestry. It also shows the modularity of development evolution often works by tinkering with the regulation of these existing toolkits, leading to huge diversity in form from relatively small genetic changes.
Okay, let's jump forward dramatically now to the 21st century. If Mendel gave us the rule book, modern science has given us, well, incredibly precise editing tools and super fast reading machines. We need to talk about the modern genetic toolkits, starting with the elephant in the room, CRISPR-Cas systems.
Yeah, CRISPR is truly revolutionary. It's essentially a programmable DNA editing system derived from a bacterial immune system.
How does it work, basically?
It uses a guide RNA molecule, which you can design in the lab, to find a very specific target DNA sequence within the vastness of a genome. This guide RNA then directs an associated enzyme, usually Cas9, which acts like a pair of molecular scissors, to that exact spot to make a precise cut in the DNA.
Like a search and replace or search and delete for DNA.
Pretty much. Once the DNA is cut, the cell's natural repair mechanisms kick in. Scientists can hijack these repair pathways to either disable a gene, a knockout, correct a faulty sequence, or even insert a new piece of DNA at the targeted location.
And the big deal is that it's precise, relatively cheap, and easy to program.
All of the above. That combination has made it explode across biology labs worldwide. It's used for basic research, creating disease models, and holds enormous therapeutic potential for directly correcting genetic diseases in patients.
Before CRISPR, though, the workhorse was gene cloning and recombinant DNA technology, still important.
Absolutely still important, though maybe less headline-grabbing now. This is the classic molecular biology toolkit developed in the 1970s. It involves using restriction enzymes, other types of molecular scissors that cut DNA at specific recognition sequences, and DNA ligases, which act like molecular glue.
To do what?
To cut out a gene of interest and paste it into a plasmid, a small circular piece of bacterial DNA. You can then put this recombinant plasmid back into bacteria or other cells and get them to produce large quantities of the protein encoded by that gene.
And this was huge for medicine.
Monumental. It's how we first mass-produced crucial therapeutic proteins, like human insulin for diabetics, human growth hormone, blood clotting factors, and many therapeutic antibodies. It basically launched the biotech industry.
So we can edit genes, we can clone them, but seeing what they do inside a living cell is also crucial.
Definitely. And that's where fluorescent tags come in. The discovery of green fluorescent protein, GFP, from jellyfish was another Nobel Prize-winning breakthrough.
You can attach GFP to other proteins.
Exactly. You could genetically fuse the gene for GFP or related proteins that glow in different colors, RFP for red, YFP for yellow, etc., to the gene for your protein of interest. When the cell makes your protein, it now has this fluorescent tag attached.
So you can watch it under a microscope.
You can literally watch it in living cells and organisms, see where the protein goes, when it's expressed, who it interacts with. It allows us to visualize cellular processes and track cell fate in real time, indispensable for developmental biology and cell biology.
Okay, these tools have fueled the rise of genomics, which is fundamentally changing medicine, moving away from just treating symptoms.
Towards a predictive, preventative, and personalized approach based on an individual's unique genetic makeup.
And the engine driving this is next-generation sequencing, NGS.
Right. NGS technologies represent a massive leap over older sequencing methods. They allow us to sequence entire genomes, or large portions of them, incredibly quickly and at a cost that has plummeted over the last decade or so.
Yeah.
We can now get a whole human genome sequence in hours or days for, maybe around $1,000 or even less in some contexts.
This flood of data enables things like genome-wide association studies, GWAS. How do they work?
GWAS are large-scale studies that look for correlations. Researchers sequence or genotype hundreds of thousands, sometimes millions, of individuals, some with a particular disease or trait and some without controls.
And they're looking for tiny differences.
They're scanning across the entire genome, looking for single nucleotide polymorphisms, or SNPs, places where individuals differ by just a single DNA-based pair. If a particular SNP is found significantly more often in the people with the disease than in the controls, it suggests that SNP, or a gene near it, might be associated with a risk of developing that disease.
Not necessarily causing it, but associated with risk.
Exactly. GWAS points to regions of interest. Often, for complex diseases, it's not one SNP, but the combined effect of many SNPs across the genome that contributes to risk. This leads to things like polygenic risk scores.
Which brings us right into personalized medicine, using this genetic info to tailor treatments.
That's the goal. For example, pharmacogenomics looks at how your genetic variants affect your response to drugs. A classic case is the CYP450 family of liver enzymes.
They metabolize most drugs.
A huge percentage, yeah. People have different genetic variants of these enzymes, making them metabolize certain drugs faster or slower. Knowing a patient's CYP variants can help doctors choose the right drug and dose, avoiding side effects or ineffective treatment.
And in cancer,
Oncogenomics is huge. Sequencing the tumor's DNA can reveal the specific mutations driving that cancer. This allows doctors to select targeted therapies, drugs designed to hit those specific molecular flaws, which are often much more effective and less toxic than traditional broad-spectrum chemotherapy.
the ultimate goal seems to be integrating even more data, not just the genome.
Right. The field is moving towards systems integration. The realization is that just knowing the DNA sequence, genomics, isn't enough to understand complex biological behavior or disease.
You need to know what the genes are actually doing.
Exactly. So, systems biology tries to integrate multiple layers of obomics data. You have transcriptomics, measuring all the RNA molecules being produced, which genes are turned on, proteomics, measuring all the proteins present and their activity levels, and metabolomics, measuring all the small molecule metabolites involved in cellular processes.
Why all four?
Because each layer gives you a different snapshot. The genome is the potential blueprint. The transcriptome tells you what parts of the blueprint are being read right now. The proteome tells you what molecular machines are actually built and active. And the metabolome reflects the actual biochemical activity happening.
It's like going from the library catalog to the books being read to the people doing things to the results of their actions.
That's a great analogy. By integrating all these massive data sets, often using computational modeling, we hope to build much more accurate and predictive models of how cells work, how diseases progress, and how treatments might affect the entire system. These integrated networks are sometimes called interactomes.
This incredible technological power inevitably opens up entirely new frontiers, but also brings some pretty significant ethical challenges. Let's explore some of these emerging areas.
Okay, on the pure engineering side, you have synthetic biology. This isn't just editing existing life. It's about designing and building novel biological parts, devices, and even whole systems from the ground up.
Like creating artificial life.
It ranges from engineering bacteria to produce biofuels or drugs, to efforts to create minimal genomes, the smallest possible set of genes needed for life, to potentially designing entirely new organisms with capabilities not found in nature. It's powerful and obviously raises safety and ethical questions.
Then there are gene drives. These sound like science fiction almost.
They are a very real and very potent application of CRISPR technology. A gene drive is an engineered genetic system designed to cheat Mendel's laws. Normally, a gene variant has a 50% chance of being passed on. A gene drive includes instructions that cause the engineered trait to be copied onto the other chromosome during reproduction, ensuring that nearly all offspring inherit it, not just half.
So it spreads rapidly through a population.
Very rapidly. The main proposed use is for controlling vector-borne diseases, for instance, engineering mosquitoes so they can't transmit malaria, or making them infertile, and then using a gene drive to spread that trait through the wild mosquito population, potentially crashing it or rendering it harmless.
But the risks of irreversibly altering an entire species or ecosystem
are enormous and hotly debated. The potential benefits are huge, but the potential for unintended consequences requires extreme caution and public discussion.
We also can't ignore the brain. Neurogenetics is trying to unravel the genetic basis of complex psychiatric conditions.
Yes, and this is incredibly challenging because disorders like ADHD, schizophrenia, autism, bipolar disorder, they aren't caused by single Mendelian genes. They are highly polygenic.
Hundreds of genes involved.
Hundreds, maybe thousands of genetic variants, each contributing a tiny amount to the overall risk. GWS and large-scale sequencing are starting to build these polygenic risk scores for psychiatric conditions.
Which raises questions.
Huge questions about prediction, stigma, diagnosis, privacy, and how we use potentially probabilistic risk information, especially for conditions that are also heavily influenced by environment and experience.
And finally, we should acknowledge that genomes aren't static islands. Genes move around, horizontal and viral gene transfer.
Right. This is a major force in evolution, especially in microbes. But it happens elsewhere, too. Viruses are constantly inserting their genetic material into host genomes. In fact, something like 8% of the human genome is thought to be derived from ancient retroviral infections.
And we've turned this to our advantage therapeutically.
We have. This natural ability of viruses to insert genes is harnessed in modern gene therapy. Scientists modify viruses, making them safe, and use them as vectors, like delivery trucks.
To carry healthy copies of genes into patients' cells.
Exactly. To replace, compensate for a defective gene causing a disease, this isn't just theoretical anymore. There are now approved AAV, adeno-associated virus, vector-based gene therapies for treating inherited conditions like hemophilia and certain types of blindness, retinal dystrophy. It's a clinical reality, albeit still complex and expensive.
Wow. We have covered an incredible amount of ground, from a monk in a garden in 1866, counting peas, to literally engineering life and delivering therapeutic genes with viruses today. What's the big picture, the final synthesis from this deep dive?
I think the synthesis is this. Mendel gave us the foundational grammar, the unshakable rules of segregation and assortment. And those rules still govern the basic transmission of genetic information from one generation to the next. They haven't been repealed. But modern genetics has provided the incredibly rich mechanism, the context, the exceptions, the regulation. We now understand inheritance not just as abstract ratios, but through the physical reality of chromosomes, the molecular details of DNA and proteins, the complex layers of epigenetic marks, and these vast interacting gene networks.
So the journey has really redefined what we mean by the book of life.
Absolutely. The genome isn't some fixed, static blueprint passed down perfectly like an immutable text. It's much more like a living manuscript.
Constantly being revised.
Constantly revised by random mutation,
Yeah.
deliberately edited by scientists using tools like CRISPR, and maybe most profoundly, constantly being annotated and modulated by external environmental influences that dictate how, when, and which chapters get read.
And that complexity, that dynamism, leads us perfectly into our provocative final thought for you, the listener. We talked about epigenetic inheritance, how things like diet, stress, maybe even exercise can create these chemical marks on the genome that might be passed down across generations without changing the actual DNA sequence. If this mechanism is real and significant in humans, how might our current lifestyle choices, the social conditions we create, the environments we live in today, how might these be subtly influencing the genetic landscape, the gene expression patterns of future generations?
It introduces this idea of a biological legacy, a kind of Lamarckian echo, perhaps, that's totally different from the purely chance-based segregation and assortment Mendel saw in his peas.
It forces you to think about responsibility in a whole new biological dimension, something far beyond those original pea plant studies.
It really does. A lot to think about. Thank you for joining us for this deep dive.
We'll see you next time.